Welcome to Berries in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries and so much more. Let's dive in.
All right, welcome everybody back to In the Interim, a podcast where we explore clinical trial science, the innovations in it, usually a statistics bend to this. I'm Scott Ferry, your host, and I'm joined by of course, none other than Dr. Don Berry. Today, we are going to talk. He's waving. For those of you on the podcast, he's waving. Some of you get visual. We actually had people, and by the way, I'd love to hear from the audience, requests, things you'd like to hear about. We had requests to go back to some trials and talk about issues that came up, barriers, how you overcame them, a little bit of maybe the sausage making, if you will. So Don and I are going to revisit an older trial, talk about the design, talk about the building of it, the different aspects of the trial design, a number of barriers in it. So this trial is a fascinating story. It's the name of the trial is the Award 5 trial, but the trial was for a drug on Eli Lilly. All of this is public. Dulaglutide was the name of the drug while we were working on it. It's a GLP-1 inhibitor.
It's a GLP-1 agonist.
GLP-1 agonist. And they were interested in running a phase 2 trial. Initially, they came to us and were interested in a 4-dose phase 2 trial for the treatment of diabetes. And the drug, the efficacy of the drug was going to be HbA1c. I'll sort of jump to the end of this and we'll come back and talk about the different aspects of the trial. We ended up helping them with the team build a seamless 2-3 trial that started with 7 doses of dulaglutide. It had an active comparator, sitagliptin, and it had placebo, so it was a 9-arm trial. The trial had a phase 2 component with adaptive randomization, favoring the doses of dulaglutide that were doing better, and we'll say a lot more about what better means.
It's dulaglutide.
Dulaglutide. We can jump to its trade name, perhaps, which is now Trulicity, is the trade name of the drug, which gives a little bit of way of whether the trial was successful. It had a component where it did interim analyses every 2 weeks during the course of the trial. During the first part of the trial, this was for response adaptive randomization over the doses of Trulicity. When it enrolled 200 patients and between 200 and 400, it could shift seamlessly to phase 3. And if it met certain criteria on the efficacy of HbA1c, but also additional parameters we'll talk about, it could move to the phase 3 components, which was fixed randomized, which could be 1 or 2 doses with sitagliptin, the active comparator, and placebo. That size of phase, the phase 3 component, the second stage of this trial, was adaptively selected. And that represented a phase 3 trial. The patients from both parts of the trial would go into the primary analysis at the end of phase 3. We controlled type 1 error as part of that. That was part of the trial design. The trial was run entirely by Bayesian algorithms. The final analysis at the end of the trial would be a frequentist analysis, an ANCOVA analysis of HbA1c. So what happened? The trial, at the first time it could possibly go to phase 3, it did. It selected 2 doses, the 0.75 milligram and the 1.5 milligram doses. moved to phase 3, enrolled phase 3. At the same time, it spawned additional phase 3 trials with the doses that were selected. The trial and the additional phase 3 trials ran out. It was a non-inferiority trial to the active comparator. It ended up showing superiority. It was very effective in weight loss, not surprisingly. The drug ended up getting approved by the FDA in 2014. In 2024, it was a $6 billion selling drug, and it's been a multi-billion-dollar selling drug since. The trial, Eli Lilly talked about saving 12 to 18 months of development time with the seamless phase 2-3 trial. So that I'm going to pause and throw it to Don to add to any comparts. So that's the trial design, this really quite elegant, reasonably complex phase 2-3 trial with interim analyses every 2 weeks in the trial, resulting in a successful trial. And we thought we would go back and revisit the trial and different aspects of the building of this trial and what happened. So Don, anything to add to the overall structure of the trial?
Yeah, I'm going to say one thing and then pass it to you for the other. And the one thing that I want to pass to you is to say something about the CUI, Clinical Utility Index, and how that came about, or maybe you're reserving that for the later time point.
I was reserving that, so we'll come to that.
And are you also reserving the DSMB?
Yep.
Because the picking the doses is there are lots of nice, interesting stories associated with that.
Yep. We'll come to that as well.
Okay, so I have nothing more to add.
Okay. All right. So we, several aspects. So what were the barriers of this? We originally, by the way, this took place, we were building this, I went back and looked, 2007, 2008-ish. We were building, so this predated even the original FDA draft guidance on adaptive designs. This was early stages of, complex innovative designs within the setting. The original trial design was meant to be a short-term, 4 months, 4 doses, 50 patients on each dose, phase 2 trial. Let that trial read out and then make development decisions.
Endpoint A1C.
The endpoint HbA1c, that's right. So as we started to look at the potential for a seamless 2-3 trial design, that trial could immediately move to phase 3, several of the initial parts that were really interesting about this were, by doing that and including those patients in phase 3, they could enroll perhaps more patients in phase 2, potentially, but also more doses. So the issue became, could we explore more doses as part of the phase 2 trial? But in order to do that, we couldn't necessarily do fixed sample size on all the doses, because then it grows linearly and now it's 7/4 as big if we're going to go to more doses. So the discussion of the number of doses and that tied to response adaptive randomization, the trial ended up, we started to explore using response adaptive randomization. The eventual trial did fixed randomization for only the first 50 patients, but then did response adaptive randomization at that point. One of the problems or one of the issues with that is you don't just want to do one of those analyses, because you want to continually learn as the data was getting more and more rich. And so now we had to do multiple analyses of this, which eventually grew to doing analyses every 2 weeks during the course of the trial.
And the definition of response became much more complicated, right?
Right. So we started to simulate this trial with HbA1c, and one of their concerns was that the higher doses could end up having really good HbA1c effects, but at the same time could have potential safety issues. And so the algorithm couldn't only be HbA1c. So at that point, it had to incorporate other endpoints into the trial. And it became clear that blood pressure and heart rate, and regulators wanted those, had to be not elevated too high for risk of cardiovascular events. So this became a big question in the design: is how do you do response adaptive randomization? How do you do dose selection when you're worried about those endpoints? And later on down the story, weight loss became a really important part to that. We can come to that. So that became sort of barrier number one, is how to incorporate these multiple endpoints in the design parameters. I don't know if you remember how this started, but one of the really interesting things that happened is we came into this and they were very against a clinical utility index initially. That they had tried to do this in some other settings and they were against it. So we started creating a number of rules that we'll take HbA1c, but if the posterior probability of heart rate increase was above some level, we would stop randomization to that. But if this was true, we would do this. And if this was true, we would do that. And we ended up creating something, and I learned the terminology at the time, a Rube Goldberg machine. And I think you referred to the design at one point as that. Do you remember that?
I do.
First of all, what's a Rube Goldberg machine?
It's a very complicated machine.
Yeah.
It's, you mentioned Post-it notes before to me. It's a very complicated machine that essentially does nothing. It just machinates.
So he created these huge things where a ball would drop, it would hit something else, a lever would fly off, 47 things would happen, and at the end it squeezed a toothpaste or something. And our design sort of became that aspect of it. And I remember you and I talking that we would show simulations and the issue was they would bring a new scenario and it wouldn't necessarily do what they wanted it to do because of the complexity of all these rules that were kind of custom to every scenario we got. We'd create a new rule, but every time we got a new one, it wasn't doing the right thing. So you and I were reviewing this. We were talking with the statisticians. By the way, Brenda Gatos at Eli Lilly was a huge hero in this, in her work and the work and the design. So you and I may talk about our discussions, but the team at Lilly was fantastic in all of this.
Just a shout out to Mary Jane Geiger, who was an MD. I mean, one of the barriers in these that we're going to talk about are MDs. And working with MDs and getting them, they're used to, they know how to run clinical trials because they've done a gazillion. And to say, well, have you thought about this or this? So Mary Jane was also critical with Brenda.
Yeah.
And the FDA was critical. We couldn't have done this without the FDA.
Yeah.
And they had some input into the clinical utility index that we're going to talk about, and we should mention that.
Yeah, and they had given feedback that they were concerned. So we talked to the FDA about potentially seamless phase 2/3, that we wouldn't have an end of phase 2 meeting where we showed the dose, but we would show the algorithm to them that would pick the dose. And they weighed in that heart rate and blood pressure were really important and gave parameters that they didn't want to see heart rate and blood pressure go above certain values. So we were looking at simulations of a number of these designs, and we saw this as a Rube Goldberg machine, and we were really concerned. And you and I thought the only way forward was going to be to create a clinical utility index, to create a weight function of these different values. That if heart rate went above a particular value, that dose had to be, pushed down. The value of that dose was lower. So we took the scenarios that they had created for us and what dose they wanted to pick. And at this point, we were probably simulating 30 different scenarios over the different endpoints. And we knew what they wanted to pick. And so we started to create mathematical functions that represented their decisions. And we thought this was the only way forward. So we went and presented them simulations of a new approach to pick the dose. And we showed them how it behaved and how often it picked different doses before we kind of jammed the clinical utility and the function on them. And they loved it. They loved the choices of this, and they became somewhat advocates for the utility function.
And so, for example, the utility was 0 if the heart rate increase was more than X.
Yep.
And the FDA, the only thing they contributed to this, except for their concern that Scott talked about, in terms of the nitty-gritty of what the clinical utility index was, is they wanted a decrease in X. They wanted a more conservative choice. And that turned out to be germane in what the doses were eventually decided.
So the utility function ended up being on 4 endpoints: the mean change on HbA1c relative to sitagliptin. So, for example, if it was within the non-inferiority boundary, that had a value of, say, 1 to therapeutic benefit. If it had better values, it had more and more values. So it was an increasing function as you decreased HbA1c up to a particular limit. And then it actually leveled off because they worried about too high of that.
And I'm sure there are some people out there that have tried to do this, and they're interested. Well, how you put these things together? You got 4-dimensional space, and are you assuming independence? Are you just multiplying the individual utilities? And the answer is, nearly. We had a multiplicative CUI, with the exception of one joint distribution or one joint function of 2 of the endpoints that were sort of the if-then that were related. So except for that, it was multiplicative.
Yep. Where if there was a parameter in the model, which was the mean change in blood pressure relative to placebo, the mean change in heart rate relative to placebo, and there was a region where if it was within a couple X, that you got full value of the HbA1c. But as those values increased, it took away from the value of HbA1c in a multiplicative way, both of those parameters. If it got above the value FDA said we don't want to see it there, it had 0 value. So it wiped out that dose as a value. The 4th endpoint that came into it wasn't really safety, and it wasn't the primary efficacy endpoint, but was weight loss. which of course we understand now plays a critically important role for GLP-1 agonists in this. But they knew that if the drug increased weight, it was actually one of the early parts that they worried if it increased weight, nobody would take the drug. And if it actually decreased weight, which they had hopes for, but in many ways they were wanting to prevent weight gain, that had additional value as a therapeutically good dose. So we showed them the utility function, and we simulated a bunch of trials and showed them the selection. And they ended up looking at these and largely agreeing, and then they tinkered with it a little bit. When they disagreed with the outcome of the trial, they changed the utility functions. They molded it around what they thought was the risk benefit of these endpoints. And it got to the point where we were simulating these trials, and when we showed them results and said, this is what happens in this example trial, they said, oh, I don't know if I'd do that. I think they'd pick the other dose. They actually got to the point where they thought they were wrong and the algorithm was right, to the point that they believed this utility function was absolutely the right way to go forward. And they instructed the DSMB eventually on that. So the utility function was then used to do response adaptive randomization over the one that had the best profile. It drove whether or not a drug would graduate and go to the second stage, whether it would bring a second dose along into the trial. And the algorithm drove all of this, which was a Bayesian model on those 4 endpoints, a Bayesian model that calculated the posterior distribution of the utilities, and it drove all of those decisions. So the one other another really interesting part to this was the longitudinal modeling within this, which we described. The original trial was maybe 4 months they were going to look at, but now the primary endpoint in phase 3 was going to be 12 months within it. And so we wanted to make our inferences based on the 12-month results for HbA1c. It was 6 months for heart rate, blood pressure, and weight loss, because that was a comparison to placebo, which they could only give for 6 months. And so we built longitudinal models of the outcomes over time that predicted the 6 month for 3 of them and the 12-month for HbA1c to drive the decision-making in the trial. Became a huge aspect of being able to make these decisions continually during the course of the trial.
So tell her about the pharmacologists and our discussion with them and what they said we should be looking at. And we said, prove it.
Yeah. So originally, we were trying to forecast HbA1c, which is a blood sugar value. They actually expected that it would be better to predict that using fasting blood glucose than using HbA1c, say at 1 month or 2 months, that you would look at fasting blood glucose. The fabulous thing was they actually had data on previous drugs. They had done quite a bit of work in diabetes. And so they were able to provide us data on fasting blood glucose and HbA1c. And so we fit the longitudinal models to the previous data. And HbA1c greatly outperformed fasting blood glucose as a predictor of HbA1c. And so we were able to abandon fasting blood glucose and use only the HbA1c values during the course of the trial. So now this is in the age of, we were simulating control of type 1 error with the regulators, with in the scenario where we're carrying forward the doses selected from the first part to the second part. We ended up sending them over 300 null scenarios to simulate, to go to regulators, and working with them to approve the seamless phase 2-3 part of it. Now, we made some concessions to the regulators on this, where at least 70% of patients had to come from the second part. They felt comfortable in that. Do you remember what Rob Hemming said when we came back to them with the updated simulations? He was at EMA at the time.
Yeah, of course. But I'll let you say it.
Okay. So we had gone to them with simulations. We had provided updates and gone back, and
they kept saying, well, what about this, and what about this, and what about this, and eventually, they kept saying no, sort of.
And we came back to them, and eventually Rob Hemming said, "We've run out of reasons to say no." And they accepted the simulation control of type 1 error. And this is round about 2008 in this scenario.
It wasn't easy in the FDA on this side of the Atlantic either. They, in their 2010 guidance in adaptive designs, had said that they were not comfortable with simulations, that they said that showing type 1 error, controlled type 1 error by simulation is not well understood. That was in the guidance. And eventually, I mean, that has the reason you said, the 300 null scenarios is because they, of course, were anal about controlling type 1 error. But type 1 error depends on, for example, what the underlying control rate is. It depends on accrual rate. And so if, as Scott indicated, if the accrual rate is fast, you may not be able to take too much advantage, and the type 1 error is affected by that. Or if it's too slow. So we had to do lots of accrual rate simulations. They became, in the next version of the adaptive guidance, which was 9 years later, they had learned to be more comfortable with simulations, and they had learned how to handle this sort of a scenario. So, for example, we've designed phase 3 trials much, since then, and we don't have to run nearly that number of simulations. We have to make them comfortable and promise them that, we'll do post analyses to make them even more comfortable, should that be necessary. And so that's actually written in the updated adaptive guidance. Just while we're on the FDA, this was at the time the critical path initiative of CEDAR dates back to 2004. But, this was Janet Woodcock's baby. And 2006, it got somewhat revised. And but this was regarded as part of that critical path initiative, even though, as Scott indicated, it predated the CID, the Complex Innovative Design initiative. And it was probably, I don't think there's been a CID that is more complicated than the Award 5 trial.
Yeah. Yep. So one of the things that Lilly did in this that was tremendous, I've never seen another case of this, is one of the concerns we had was we have this algorithm doing response adaptive randomization. It was using longitudinal models, so it needed somewhat mature data to do a good job at dose selection. Is that we simulated the trial under a range of scenarios. They're concerned that you send this out to operations and they enroll gangbusters, which you can do in diabetes. So we simulated a range of scenarios, and we looked at the likelihood of making good decisions, picking the right dose before it went to phase 3. And Lilly, they created boundaries, and they said, we have to stay in these boundaries. And they did not want to enroll more than 8 per month, for example, because they knew that it was going to make worse decisions. And so they told operations, you can't go above this number, which I think was a huge part of the success of the trial.
Yeah, and so that made the trial somewhat longer, but nowhere near the savings that you indicated earlier on in terms of, but this was taken into account in their calculation about the savings.
Right. So now, as you described this is a reasonably complicated design. It has got a clinical utility index. It's got interims every 2 weeks. The algorithm updates the randomizations. It's got prospective go to stage 2 rules. So now we start working on the implementation of this, the operations of this. And
along with that, it also picked the sample size for stage 2. And the sample size it picked when it eventually went was the smallest possible sample size. And it was a minimum number in stage 1. It made that decision in the actual trial as soon as it was allowed to make it. And at some point, I want to tell you about the DSMB and working with the DSMB.
Yeah, so let's shift to that. Let's shift to that. So they created DSMB, and we spent a lot of time showing them simulations of the trial designs, explaining the utility index, the decision rules. And Lilly told the DSMB they weren't allowed to change the decision of the algorithm around doses. And it was how much they believed in that utility function to be the right dose. They could stop a dose for safety during the course of the trial. They could stop the whole trial for safety. So that was still a huge part of it. But they knew simulation control of type 1 error was a big part of the design, and the utility index was a big part of it. And so the DSMB was fully on board with this. So you and I were advisors to the DSMB, and we were unblinded. And if you remember, these would happen every 2 weeks on Wednesday, every fortnight on Wednesdays. These reports would come out. So a number of really interesting aspects to the DSMB, which have been made public. So yes, jump into that.
So you said they were on board. They were more than on board. They said, you've told us that we can't change the design. We can't drop an arm for lack of efficacy. We have to follow the design. But if we were making all of the decisions, we would make exactly the decisions that the algorithm has made. And the algorithm, didn't choose the highest doses, even though weight loss was, incredible on the highest doses. And it toward the end of that period, when they could drop a dose for safety, they were worried about the highest dose. And they understood, they were actually tracking things and, how things were going and how close to making a decision to pick this dose over that dose. And so they were worried about the highest dose. And I said, you can drop it now. If you don't drop it now, and next week when we get, the permission to pick the 2 doses, if you dropped a dose that would have been picked, we still go with 2 doses, according to protocol. If you wait until next week and the highest dose is one of the ones that's picked, and you decide to drop it then, we continue on only 1 dose. So they understood that. And so they hastily dropped the highest dose for safety. And it was, the combination of blood pressure and heart rate. Considerations that, by the way, these doses are now part of the marketed. The marketed doses were the ones selected in these phase 3 trials. The other phase 3 trials were the same ones, and they were happy with that. And, we got the right dose. But they learned over time that heart rate, even though it was increased, was not that important and didn't have clinical ramifications. And so the marketed doses now include higher doses than, one dose that's higher than the ones even that we considered in the Award 5 trial.
One of the interesting things is when they dropped the high dose, it had already been zeroed out in the randomization by the RAR. So it was
it was zeroed out when they were allowed to zero out, right? For the final analysis. But it was predicted based on what the utilities were. You could see that, almost certainly next week it's going to be the same, and they're going to drop it. It's going to be dropped.
Yep. So and we could see, and it was the results were amazing, that when it reached that 200 minimum, it was going to jump at that point. So it was one of the designs where the design ran exactly as planned. The algorithm ran, the RAR was updated, the decision was made, the DSMB agreed with each of the decisions in the course of it. And the trial picked 1.5 milligrams and 0.75. And one of the interesting parts is neither one of those doses were part of the original 4-dose phase 2 trial. So at the time, this 1.5 milligram and the 0.75 milligram were, really good doses in terms of this, and they may never actually have been used with the other phase 2 trial. So it had a number of aspects, sped up development. And by the way, then it jumped to phase 3. Those doses moved on and into the other trials, and hence, going back to the start of the story, it ended up showing superiority to sitagliptin, won all of its phase 3 trials, showed benefit in its cardiovascular risk trial, and is now Trulicity.
And Trulicity is the initial GLP-1 agonist. You read today about the other diabetes drugs that are GLP-1 agonists, but the big deal is the weight loss. And the weight loss, by the way, in the Award 5 trial, in terms of the doses, was completely predictive of the weight loss in a later trial that they ran with Trulicity. That showed that the dose by dose, it was almost this, matching what we would have predicted from the Award 5 trial. But Trulicity is not approved. Lilly hasn't looked for it to be approved for weight loss because they have other GLP-1 agonists that are, next generation that are even better.
Yep. So if you'd like to read more about this, you can read about the Award 5 trial. There's paired publications about it in the Journal of Diabetes Science and Technology in 2012. Publications of paper before the results came out of this. You can read those. Want to acknowledge Zach Skrivanik, Jenny Chen, Mary Jane Geiger, who Don mentioned, Andy Anderson and Brenda Gatos, who are on those papers as well, doing huge work and worked with them on the team. And this was about 9 months that we built this trial and got it ready to execute.
Just one additional aspect of the 9 months. Took us 9 months, but I started to consult with Eli Lilly in the 1970s. And we built, Scott and I, over time. I don't know if Scott was pretty young in the 1970s. But when he became old enough to go to Indianapolis, he went to Indiana. And we developed a really great relationship. And we couldn't have done this without the support of the statisticians, all the way back to Charlie Sampson, who set up biostatistics at Eli Lilly, and also was a co-founder of the Muncie meetings, for those of you that might have been around back then. Muncie, Indiana, where Ball State University is, and I presented their adaptive designs because of Charlie and others at Lilly. But and they designed, this was Prozac days, they designed adaptive trials in Prozac. So they, shout out to all of them. If you pick a random pharmaceutical company and drop us by parachute onto their campus and see if we can persuade them to do this kind of thing, I'm not sure of the results, but it's not a certainty that we would be able to do it.
Yeah.
Because we don't have the trust build up and the great working conditions. So it was all a great story for us. And it was a great story for Eli Lilly, who decided that we got to simulate every trial. Yeah. Every trial that we run, we're going to simulate. And I don't know whether they're still doing that, but it's not a bad thing, I'll tell you.
Yeah, no, Karen Price is there now with many other statisticians, and they're still simulating. And by the way, they're the largest financial pharmaceutical company in the world at this point, I think. All right. Well, thank you, Don.
I don't think that's true, but they are certainly the biggest on Wall Street.
Yeah, their market cap or something. Yeah. Yep, that little Indiana company. All right. Well, so thank you, Don. We're going to jump into some interesting trials as we go forward. And again, if you have any ideas for the show, holler at us. In the meantime, we are in the interim.
Yeah, and if you have questions about what we've said or what we did, please, send an email.
In the interim, thank you, everybody.
Thank you. Thanks, Scott.
Bye.